It's the tiny organism behind our fluffy bread. But now, this organism, baker's yeast, might also be pointing us towards something far bigger, the possibility of life beyond Earth. In a fascinating new study, scientists have found that yeast can survive conditions similar to those on Mars. To understand how this humble microorganism could endure such alien extremes and what that means for the future of astrobiology, I am joined today by Dr. Purusharth I. Rajguru, Associate Professor at the Indian Institute of Science, Bangalore, and corresponding author of this study. Welcome to our channel, sir.
Yeah, thank you, Dr. Jain, for having me. It's wonderful to be talking to you.
Yeah. I am Dr. Anubha Jain, Senior Journalist with ETV Bharat, and I will explore this incredible journey of yeast from the kitchen to the cosmos. So, sir, first I like to know what inspired your team to test baker's yeast under Mars-like conditions, and was there a specific discovery or curiosity that sparked the idea of combining shock wave physics and cell biology?
Yeah, I've been working with yeast since 2007 now, so it's been close to 20 years, more than that, I guess, right? That was during my postdoc when I started working on yeast. And even though this is a very simple unicellular organism, the beauty of this organism is that whatever you learn in this organism is often found to be conserved in you and me, right? So it's true even in humans. And that's how we have learned so many things about us by focusing on this model organisms. It's not surprising. That's why that many Nobel Prizes have also been given to work that was, to begin with, done using Saccharomyces cerevisiae, which is the scientific name of the common or the baker's yeast that we use in our kitchen, like you were pointing out. So as a system, it's fantastic to work with. When I moved to Indian Institute of Science, Bangalore, Department of Biochemistry in 2013, I decided I wanted to continue working with yeast, and that's what we have been doing. Of course, as a part of evolution, we have added more model systems to our repertoire, and we continue to do that. But yeast is a very important aspect of whatever we do in the lab. So even before we did this work on Mars, we have been doing many experiments with yeast, and we have published papers on that. Regarding why we looked at Mars-like stress conditions and we decided to use yeast, 3 years ago when this idea came to my mind, the motivation was money, surprisingly. I say that because we had some grants and the funding, we were waiting for those funds to be released, and so there was a situation where there was a bit of a vacuum. We were trying to get hold of some money, and at that time I saw this call that said that the pot of money from ISRO is not very big, but it will be something to explore something new, which will be related to space and astrobiology, and that got me going. That we have this wonderful system, and nobody has looked at it in context of astrobiology, in context of Mars and Mars-like conditions. So we decided, why not try it and let's present this idea. And I was fortunate that ISRO, that is Indian Space Research Organization, they have a space technology cell at IISc. They liked the idea, they funded it. And I got a very good collaborator in form of Dr. Balamurugan Shivraman, who is also one of the authors from PRL. And we had a fantastic project assistant, Ria Dhage, who did all the work. And that's how things worked out. So the motivation was, to some extent, how can we get some quick, small funding? But at the same time, we wanted to try something that was not tested earlier. It was out of our comfort zone, and it just clicked.
Yeah, no doubt. It's a very unique concept. And so how difficult was it to expose live yeast to such extreme shock waves without destroying them completely? And also, could you describe the challenges of setting up and running the high intensity shock tube for live cell experiments?
Yeah, so from our literature search, we did not find any evidence of yeast being exposed to shock waves. So that was a first. Like you said, we had to ensure that it got shocked and didn't die, right? I mean, you can bombard them, but after that, if you're not going to get any live cells back, then you can't do any study. So the idea was to find a shock wave condition that was sufficient to bring about a physiological change in the yeast cell, but not kill it.
Mhm.
So the shock waves are one of the expertise of Bhala Murugan Shiv Raman. So they had it in Ahmedabad. Ria Dhage, who is the first author, she worked very hard to basically troubleshoot this. Like I
Okay.
said, there was no protocol to follow. So she had to do lots of trial and error runs where she would expose the yeast cells, check how much is the survival rate. The other problem is that when you expose these things, you can't do it in aseptic conditions, which is what we normally do in the lab. So we have specific instruments where we culture these cells so that they do not get contaminated with other kinds of cells. This is very
True.
important because you only want to be working with your organism of interest. But in PRL, there are no biology labs, and in Bala's lab, we didn't have any such instrument. So Ria had to basically make sure not only that yeast survived, but it also didn't get contaminated with other things. She also managed to get some help from IIT Gandhinagar in terms of doing the experiments. So we were fortunate in that sense.
Mhm.
And troubleshooting took a while, but at the end of the day, I think Ria managed to find a condition where we were able to actually shock the cells and still able to recover enough cells after shock with which we could do downstream experiments.
Mhm. Okay. So, what surprised you most about the East response to these harsh conditions? Any such scenario you faced?
Yeah, so we didn't expect yeast cells to survive the intensity of the shock waves that it eventually did.
Mhm.
In the paper, we observe that shock waves of a certain intensity, which is 5.6 Mach. Mach is a unit of measuring these shock waves. Yeast were able to survive that. And beyond that, we couldn't test because Bala said this is the maximum intensity we could generate in our lab in the current conditions.
Okay.
So we were very surprised that after that, even though only a certain percentage of cells survived, not 100% survival was observed, but some cells did. And that's perfectly understandable because when you give any kind of a shock, you will see some amount of cell death. It was heartening to see that not only the cells survived, but after a brief lag period, they were as good as what they were before the shock, right? So it's
Okay.
almost like you fall sick and then you recover out of it, and then you are as if you had never fallen sick. So
Mhm.
this resilience of our baker's yeast is what really surprised us, and that makes us hopeful that if there was an extinct life form, or if there is an extant life form, then there could be life like yeast that perhaps could have lived once, or who knows, might live in future.
Mhm. So, what does this study tell us about the possibility of life surviving on Mars or other planets?
Yeah, so before I get into that sort of a conjecture, a clear
Mhm.
disclaimer here that what we have done here, Dr. Jain, is entirely in the laboratory.
Mhm.
The shock waves that we have generated, there's also a perchlorate stress that we have used in the paper in our study. And this sodium perchlorate is one of the salts that's present on the Martian surface. We know that.
Mhm.
And that's also a highly abrasive chaotropic agent which kills life forms because it destroys the proteins which are important in our cells. Both of these conditions are that have been tested within the lab, right? Uh, we have tested them individually and we have tested them together. But this is only two of the so many different conditions that one would come across either on Mars or any other planet, right? So it's a very good start, and we could say that as far as these two conditions are concerned, yeast seems to be able to survive. But I would add in the same breath that when you are faced with several such, shocking conditions or unpleasant conditions for any life form, it's hard to know what will happen, right?
Mhm.
So that's where the next part would be, or the next episode of this study would be to try and combine more such conditions. For example, temperature temperature is very different on Mars, gravity is very different on Mars. They have microgravity conditions. Now, can we start combining these conditions with what we have already tested, and we make it more complex conditions and then ask, could yeast still survive?
Mhm.
Uh, that needs to be done. But as far as these two conditions are concerned, the answer is that yeah, it's able to survive.
Mhm.
So it makes us more hopeful that we could do these experiments with yeast and, of course, with other organisms as well, and go along these lines of experimental testing. That's a big positive that has come out of this study, that yeast will be a good model system to look at this in the context of condensates that we were looking at,
Mhm.
and one should do more such experiments.
Okay. So, you mentioned in the paper that the role of stress granules and P bodies in helping yeast survive. So, do you think similar mechanism could exist in other microorganism or even in higher life forms?
Yeah, that's a fantastic point you raised, Dr. Jain. And that's in some ways is perhaps the most interesting finding of the paper. Of course, yeast surviving these conditions is a good indication. But I think what we have gone ahead and done is to actually identify a tool that can be looked at not just in yeast but in other organisms, and that could give us an idea about the survivability of these organisms. So stress granules and T bodies, just for the for viewers who may not be aware, these are basically RNA protein complexes that are present in almost all model organisms that have been looked at. So they are present in single-celled yeast, and they're also present in you and me. So right now in our body, lots of cells will be having will be having P bodies and stress granules. These RNA protein complexes, they decide the fate of the mRNA. And mRNA is something that comes from the DNA. It's an intermediate to the protein synthesis aspect, right? DNA gives RNA, RNA is then converted to protein. This is fundamental biology. The RNP condensates, also known as RNA granules, these structures often decide whether these mRNAs will get translated or not. And they are very active in response to stress conditions.
Mm.
And what we have identified here is that here's another condition which is stress for life forms. And these RNP condensates are forming under these conditions. And when you create a condition where a cell cannot form these condensates, the cell is not as good as in its ability to deal with the stress as the one that can actually form these granules or condensates. So it has provided us a tool which, even in other organisms, for example, it could be a tool to look at the health of the astronauts. So one could measure the assembly of these condensates in blood cells of the astronauts, and try and look at what is the response of this astronaut to a certain kind of a condition that they would come across when they are on board, or when they are getting exposed at space stations and things like that. So that is another very important learning that has come out of this, that condensates could be a marker for some of these stress factors that we would come across when during space travel, or even during your stay on space stations and things like that.
Okay, right. So, how could this discovery influence future space missions, or are there any plans to send yeast or similar microbes on upcoming space missions to test their survival in real conditions?
Yeah, so yeast has in past been sent to space, right?
Mhm.
So that's why I was mentioning that yeast is not only a great model system for regular science projects, biology projects that we all do in our laboratories worldwide, but they have also been found to be a good model system for astrobiology related projects. NASA has sent yeast in the space. What I would like to think now is that our study should hopefully convince people that in some of the missions that would go from India, we could have yeast on board, and we have a proof of principle indicating that, so you can only come up with so many conditions in the lab, right? Ultimately, the proof of the concept would be that you actually expose the yeast to space or let's say Martian conditions, and then ask the question that, what is the status of these RNP condensates? How is its physiology changing? Is it able to cope up or not? Right. So I'm really hoping that this will be able to convince people that even though yeast is a very simple organism, I think this study should galvanize efforts of having yeast on board for sure. But at the same time, our study is a proof of principle that RNP condensates like stress granules and P bodies would be a good tool for many studies related to health of the astronauts, as well as understanding survivability of not only yeast but different model organisms in space explorations, as well as, for example, India is planning to come up with its own space station,
Mm.
right? Therefore, a lot of different experiments will be possible once we have these space station of our own. So I'm hoping that we will be able to do similar kind of experiments, and as a lab, as a group, we are really excited to contribute towards that.
Okay. So, what are the next step for your research team? Your concluding note.
This is just the beginning, right? Like I said, this is the first time we tried this idea, and I'm glad that it clicked. Although to the viewers, I would like to say that scientists are not always lucky like this. You often come up with ideas, and you tried once, twice, thrice. Often things don't work out, but thanks to the resilience of the scientist, we continue to keep trying. In this case, we are lucky and it has worked and it has given us a proof of principle. So I think, obviously, like I said, combining other Mars-like conditions or other space conditions to whatever we have learned, and addressing this question in the context of survivability as well as the status of RNP condensates is something that we can do in the lab, and we would like to do that. But eventually, I think it will be very nice if we could do these studies as part of future space explorations, where we can send our yeast. And the good thing here is that since we have been studying RNP condensates for past 10, 15 years, we have all the tools in the lab. These are genetic tools. What I mean by that is these are strains of yeast which are mutant for certain specific factors which are important for RNP condensates in different forms. So when you have this whole battery of tools, you can not only simply understand survivability and observation based, you cannot just do observation based experiments, but we are actually quite ready to actually come up with mechanisms that could be underlying how is it exactly happening. We have done to a certain extent that in this current publication, but again, that's only two conditions that we have looked at. When we are looking at several conditions, or for example, if the yeast ends up actually going on these space explorations, we could have all these mutants that we can expose them to these conditions and then ask, how is the cell physiology changing? What are the RNAs that are increasing in numbers? How is the conversion of mRNA to the protein changing under these different conditions? And that information, I think, will be extremely useful in eventually understanding how does our body, in how does a life form respond to these kind of stress conditions, which is what we really need to understand for future explorations.
Right, right. And thank you so much, Dr. Rajaguru, for sharing your fascinating insights. And this remarkable study reminds us that even the simplest form of life, like the yeast in our kitchens, can teach us profound lessons about survival beyond Earth. So, from baking bread to braving Mars, this tiny organism could help pave the way for future space research and missions. Who knows, the next time we send the astronauts to Mars, yeast might just be part of their crew. Thank you so much. Thanks a lot.
Yeah, thank you, Dr. Jain, again for giving me this opportunity to share whatever we have learned with your audience and viewers. This is something as a scientist I always look forward to. We publish our papers, but if we can convey the crux of these studies with common man and woman, then I think that gives us lots of satisfaction. So your portal is giving me this opportunity. So thank you very much.